Akanksha, can you hear me? अच्छा खासा वहां से हो रहा था. Akanksha, Akanksha, do you hear me? Akanksha, Akanksha Yadav, please share your screen. We are about to start. Akanksha. Akanksha, you are not audible. Please unmute your mic.
Yes, sir.
Yeah, you are not. Yes, you are now audible.
Okay, sir.
You can start your presentation in a running mode, please.
Okay, sir.
Wait a minute. First of all,
Good
I would like.
afternoon, sir.
Wait a minute. First of all, I would like to welcome our session chairs, Mr. Yogesh Pal and Mr. Saurav Srivastava, sir. And Mr. Yogesh Pal is, yeah. So Yogesh Pal, he's from Bennett University, Greater Noida. And Dr. Saurav Srivastava, he is from Moradabad Institute of Technology, MIT, Moradabad. I really welcome the session chairs and I really thank them for being with us, for sparing some precious time for their schedule. Thank you so much, sir. So with your kind permission, can we start the session? Okay, okay. Now, Akanksha, you can start your presentation.
Okay, sir. Good afternoon, everyone. Myself, Akansha Yadav. I'm presenting my paper presentation. My topic is leveraging deep neural network for early recognized lumpy illness in animals. These are the content I'm using in my presentation: introduction, proposed model, result, and discussion conclusion. Now start the introduction part. Lumpy skin disease is an emerging bovine viral disease. All ages breeds of cattle are affected, but especially the young cattle in the peak of lactation. The common symptoms of this disease are fever, infertility, reduced milk production, poor growth, and sometimes death. The animal processed high fever, which is followed by the development of large nodules ranging 5 centimeter in diameter. LSD is characterized by the appearance of dermal lumps of varying size, which can be quite painful for the affected animal. These lumps typically manifest on the head, neck, and limbs of the cattle. Lumpy skin disease affected both agriculture, economical, and animal global, globally, making livestock a major danger. Okay, this is the taxonomy of lumpy skin disease virus. Basically, this figure is saying this climate change is increasing the number of fly population increase, which affect sorry, which affected healthy cattle, infect them and then separate the illness of their calves through milk. Infected pregnant cow can also transmit the disease on their fetuses. This highlights the cascading effect of climate change on disease transmission in cattle, as it increased the likelihood of likelihood of outbreaks through the insect vector and subsequent transmission mode. Lumpy skin disease virus is transmitted from infected to non-infected animals through the direct contact by mosquito or flies and ticks, maybe the environmental variables. Because of the disease is affected on reduced milk production, weight loss, miscarriage in pregnant animals. Damage to hides and occasionally even death. Farmers and the cattle sector as a whole suffer large losses. One-
Akansha, please make it fast.
Okay, sir. Okay, sir. Methodology used in this paper dataset: lumpy skin, two classes: lumpy skin cows and normal skin cows. Then image augmentation, and then splitting dataset, test train and validation. Model we are I'm using VGG16, VGG19, and CNN. Then result. Utilize a Kaggle dataset for lumpy skin condition consists of image and data collected from several veterinary clinics, collected by specially trained medical personnel, connected 10-20 images of both healthy and sick cattle before 700 images for normal skin cows and 320 images for lumpy skin cows before augmentation, and contains the 50-50 images of both healthy and sick cattle after augmentation. This is the sample images of dataset. And CNN and VGG16. My proposed model is VGG19. VGG19 is deep convolution neural network architecture with 19 layers, known it is accuracy in image classification. We utilize the VGG19 model, a deep convolution neural network renowned for its image categorization proofs. Training it for 30 posts. This process allowed the model to learn and adapt to photo character size. Its superior performance, surpassing other models, underscores its depth and design, effectively capturing infected patterns and nurses. The model's robust performance and demonstrated its ability to learn generalized from the training data, highlighting its efficiency, accurate photo identification. VGG model VGG19 models result basically show the result prediction image VGG19 model predict lumpy or predict normal. Accuracy of this model. VGG90 performs is exceptionally well, achieving the highest accuracy of 96%. In comparison of existing model and accuracy, CNN I got the 85% accuracy, and VGG16 I got the 95% accuracy, and proposed model is VGG 96% accuracy. Conclusion: there has been a lot of research on the character size of human skin diseases. Have been a few study on the classification of skin disease in animals. In this work, we created a technique to identify and categorize the animal lumpy skin disease as normal, mild, and severe. They introduced VGG19, a deep learning model for lumpy skin disease image classification. We used augmentation techniques in order to improve the dataset and evaluate the model's performance after training. More accuracy is 96%.
Okay, very good. Uh, Tell me the basic difference between BGG 16 and 19. Both are transfer learning approaches, no? So tell me the difference
Yes,
between
sir.
BGG 16 and 19.
Okay. So, the VGG 6 layers difference in VGG 16 and VGG 19 model, basically.
Huh?
Yes, sir.
How many layers in BGG 16?
In VGG 16 model have 16 layers, and VGG 19 model has 19 layers.
Okay. Uh, Difference
convocational.
between CNN and VGG 16.
Yes, sir.
Difference between CNN model and VGG 16.
Okay. The
Okay, good.
same model 16 and CNN.
Huh? Now both are
Same.
different.
Sir, architecture is a the VGG 16 is a deep convolutional neural network designed for image classification task. Both.
The purpose of both classifier is same. Okay,
Yes,
image
sir.
classification,
Yes.
but the
Yes.
architecture are different. Okay,
Sir,
good. Hello.
Thank you, Akanksha. Thank you for presentation.
thank you, sir.
Next, I would call upon Parisha. Are you there?
Yes, sir.
Can you share your screen?
So please allow me to share.
Okay, wait. Arey, mouse. Now you are allowed. Please share your screen.
Yes, sir. Yes, sir.
Yeah, okay.
So is my screen is visible?
Yes. Press F5. You can zoom in your slide.
Yes, sir.
If it's not happening, please zoom in your slide. Now it's okay.
Yeah.
Okay, please.
Good afternoon, everyone. My name is Parisha, research scholar from Babu Banarsidas University. The topic of my paper presentation is improving cardiovascular forecasting precision with blended machine learning methods. Here are the contents: introduction, methodology, proposed model, result and discussion, conclusion, and references. So, start with the introduction. So, using machine learning to classify cardiovascular disease occurrences can help diagnosis, reduce misdiagnosis. So, this research develops a model that can correctly predict cardiovascular disease to reduce the fatality caused by cardiovascular disease. So, the heart attack and chest discomfort can result in the cardiovascular valves narrowing, which causes the supply of blood to the heart to slow down or stops. So, various specialists or extensive training, the expertise needed to diagnose cardiac conditions, and the goal of this extraction is to derive the knowledge of significance from massive data sets. So, large amount of health information may be used to create hypotheses and to retrieve the issues, health-related information. And the challenge of categorization is used to identify unknown samples in the variety of situations. And the goal of this research is to create effective algorithmic methods for classifications. So a system that uses the categorization will let the doctors analyze patient and determine the likelihood that they are at the risk of developing the disease or the cardiovascular disease. The various researchers like Lenson and Nielsen in 2005 uses the naive base. So in our research, we will compare the naive base with the hybrid naive base and find out the conclusion. We have methodology. We have used a machine learning algorithm that is naive Bayes and hybrid naive Bayes. So in naive Bayes, it is used for finding characteristics that may be identified and utilized to differentiate between cardiovascular sufferers and people in good health in the basis for categorization and data analysis. The classifier is a basic variant of the Bayesian network classifier, which relies on the robust interdependence for of variable assumption and applies the Bayes theorem. The often used approach naive-based categorization because of its ease, effectiveness, and strong collection of data quality. But the thing is that naive-based performs poorly on the set of information containing complicated variable relationships. So huge amount of data will yield a reliable result using naive-based classifier. So there's a correlation between characteristics and health issues in the medical job. So the hybrid version of the naive-based classifier is listed as a solution or its shortcomings. Hybrid naive-based in terms of feature independence. Hybrid naive-based categorization performs better than naive-based and comparable to a Bayesian algorithm, which avoids unmanageable complications and consider the effect of every features. The structure extension based hybrid naive-based method requires additional instructions time because it uses the every single data at the time of the data analysis. And for the coronary illness data set with interdependent characteristics, we use hybrid naive-based since there is an increasing demand for implementing techniques from data mining to health information mining. Next, we have proposed model. So we obtained the cardiac STELOG data set for our investigation from UCI repository. To conduct the research, we use primarily processing method, which is below, that is replacement values that are missing, separation, interquartile ranges. Replacement values that are missing is used to every MPT characteristics values that employed replacement values or that are missing. So the more the mean from the initial collection of data are used to this filter and to fill the values that are missing. Separation is used to utilize unattainable separation, and that is separation filters that were used separating numerical characteristics. And interquartile ranges is filtering a variance or measurement. The information collection is split into various intervals by it. So we have Q1, Q2, and Q3. Q1 is initial halls of a ranked order gathering of information. That is median number. Q2 is an information collection, that is mean value, and Q3 is an amount that represents the center portion of the information collection. Next, we have result in discussion that we use naive-based categorization using Vector 6.4 for our research. We made use of cardiovascular information obtained from UCI. There are 270 occurrences and 14 characteristics in our cardiac STELOG data set, validation across test and used to evaluate hybrid naive-based. So here there are two tables, that is 4.1 and 4.2. 4.1 displays the characteristics from the cardiac data collection, that is cases 290, and the quantity of the features is 15. In our second table, that is 4.2 heart disease data set, which provides the information for the data set, that is test conducted 30 and training is 315 on instances, that is cross validation on 20 folds. So our investigation included measurement of precision, sensitivity, specificity, and predictive power to access hybrid naive-based effectiveness. So certain indicators of performance which are generated using ambiguity matrices are particularly significant in the healthcare industry. So these are the ambiguous matrices, that is real favorable, untrue favorable, sometimes it is inaccurate favorable, and real unfavorable. These are the predictions according to ambiguous matrix. These are the subsequent categorization matrices that are based on ambiguity matrices. That is table 4.3. Effectiveness of favorable prediction, that is real favorable or inaccurate favorable with the help of real favorable. And if we estimate the estimate worth of unfavorable, that is real favorable, unfavorable, or untrue favorable with the help of real favorable. So whenever the testing is favorable, the EPF, that is effectiveness of favorable prediction, estimates the likelihood that the illness is prevalent. Or when the examination yields an adverse outcome, the EWU, that is estimated worth, that is unfavorable, defines the likelihood that the illness does not exist. So here there are some of the table, that is 4.4, 4.5, and 4.6. So for table 4.4 documents the accuracy of the data set or efficiency of a cardiac STELOG collection, that is the technique that we have used, and the precision. The technique that we have used is hybrid naive-based with interquartile and precision in 98.98. In our
Please conclude your presentation, please.
Okay, sir. Okay. These are the tables. So here our conclusion. In this present research, we used hybrid naive-based classification to diagnose cardiac conditions, and we examined the cardiac STELOG data sets effectiveness using hybrid naive-based. The result of the research indicates the hybrid naive-based approach performs better than each of the alternatives that we have used or we have seen in various research papers. The suggested method uses IQR, that is interquartile filtration and separation, to increase hybrid naive-Bayes performance up to a 98.98% effectiveness. That was greatest approach suggested in comparison to naive-Bayes categorization. So this model automated illness identification and with the dependent characteristics for illness evaluation. So yes, hybrid naive-Bayes classification is a potential framework for medical information sets such as cardiovascular disease. These are the references.
Okay, ma'am. Uh, Tell me how the HNB is different to compare than NB.
Sir, actually, the occurrences of the certain features that is independent of the occurrences of the other features, such identified on the basis of like colors, shapes, taste, or the area that is aerial or spherical, that is recognized as an entity or an object. So each feature individually contributes or liable to identify, that is, it is without depending on each other.
Okay, thank you.
Thank you.
Thank you, Parisha, for your presentation.
Thank you, sir.
Next, I would call upon Vaishnavi Yadav. Vaishnavi,
Hello, sir.
yes, Vaishnavi, you
I'm
can.
sharing my screen.
Yes,
Yes, sir. Is it visible?
yes, it is visible. You can start your
Okay, okay.
presentation.
Yeah. Good morning, sir. My presentation is advanced deep learning techniques for enhanced this content. Introduction. Eyes are the essential part of the human body. Each and every person relies on the eyes to see and sense the world around them. It is one of the most vital sense, eyesight, because it accounts 80% of all the information we take in. With the help of the retinal images, we can identify some eye diseases. To accurately diagnose eye illness, it is required to analyze a wide range of symptoms. Globally, at least 2.2 billion people have near or distant vision impairment. In at least 1 billion, or almost half of these cases, vision impairment could have been prevented or yet to addressed. These 1 billion people include those moderate or severe distance vision impairment, blindness due to unaddressed refractive error, cataract, eye, age-related macular degeneration, glaucoma, diabetic retinopathy. These are the data of the WHO, as well as near vision impairment caused by the unaddressed psychopathy, 8.26 billion people. This is the proposed methodology I'm using in this paper. I collect the multi-class data set, then split into a train testing and validation in the ratio of 70, 20, and 10. Then I used the different models like VGG19, VGG16, GAN, and CNN. And then I got the result. To validate the result, we tried different models on my data set. Then I'm using CNN, convolutional neural network, and generative adversarial network, GAN, and VGG 16 and 19. So I got the best result from the VGG 19. And I am using the data set, collect from the different variety of sources like ocular recognition, diabetic retinopathy, image data set, and all. So I have a data set which are 4 classes: cataract, glaucoma, normal, and diabetic retinopathy. Each class have around 1000 images, like cataract has 1038, glaucoma has 1007, normal is 1074, and diabetic retinopathy has 1098. These are the sample images of the data set. Now, these are the results. I have using 4 models, which is 16, 19, GAN, and CNN. So these are the results I got from the different models, which is 16 has 17% accuracy, GAN has 54, CNN has 86, and which is 19. My proposed model has got the best accuracy, 96. Each model is completed 100 epochs of training to reach the best performance. Maintaining a balance in each value is crucial for achieving the optimal model performance. So, training for VGG19 is terminated at 53 epoch due to lack of progress observed after 10 modifications in the learning rate. The training lasted for an hour, 55 minutes, and approx 4 seconds. These are the training time, and this is the training and validation accuracy and loss graph I got from the my proposed model. It shows that the accuracy of a training and validation is optimal at epoch 35, whereas the loss of the training and validation is optimal at 24. At 35 and after 24, it will in a same parameter, like it will not increase. The categorization finds quite good with the model accurately recognizing 4 categories. It will shown in this figure. Uh, Confusion matrix of my VGG19 proposed model. It will show all the classes, cataract, glaucoma, diabetic retinopathy, and normal. The matrix entries indicate the number of data points for each combination with the actual labels as rows and the prediction label as columns. These elements help in the computation of several assessment matrices that determine how well the model performs in the multi-class classification tasks, including accuracy, precision, recall, and F1 score. These are the evaluation parameters I got with the help of this confusion matrix, like precision, recall, F1 score, and support. These are the parameters, and I got the 96% accuracy with this model. Conclusion. This that the deep learning algorithms are used for retinal images to effectively diagnose retinal illness such as normal cataract glaucoma and diabetic retinopathy. The VGG19 model has high training, validation, and testing accuracy. 92% of training accuracy, validation has 96, and training has 93. Our approach to identify the retinal abnormalities is very accurate and has the potential to enhance patient outcomes with early detection. However, further clinical studies are needed to confirm its usefulness. Further research might to improve the performance of the proposed classifier by experimenting the different models and adding more pictures of classification. Thank you, sir. This is from my side. If you have any questions, please ask.
Hello. Thank you, Vaishnavi.
Yes, sir.
Tell me the difference between precision and recall.
Yes, sir.
Difference between precision and recall. Why we calculate precision and recall?
So we we calculate precision we calculate the precision and recall for know the better results of our research or model, like in
Okay. True. What is true positive, false positive, true negative, false negative?
so true negative yes sir, true negative is a abnormal cell classified as normal, and false positive is like misclassified as abnormal, and false negative is like misclassification, and true positive is like its actual prediction. It's like the image has cataract and it's predicted as a cataract.
Okay. Thank you.
Okay, thank you, sir.
Thank you, Vaishnavi. Next, I would call
Yes, sir.
upon Vibhor. Vibhor, are you there?
Yes, sir.
Vibhor? Vaishnavi, you can leave the meeting.
Okay, okay, okay.
Vibhor? बोल चले. Vibhor?
Yes, sir.
You
I
can
am
share your
available now.
screen. You can share your screen.
Just sharing my PPT, sir.
Yes. Vibhor, please make it fast.
Sir, is it visible?
You have to share your screen, not the window.
Sir, my PPT is visible.
No.
Okay. Is it visible now, sir?
Yes, it is visible.
Okay. Good morning, everyone.
Please press
I
F5.
Vibhor Kumar Vishnoy. Here I am to present my paper titled Fine-Tuning the Pre-trained Convolutional Neural Network Models for Plant Disease Detection Using Transfer Learning. These are the outlines of my paper: introduction, literature review, proposed method, data set description, followed by disease classification.
Slide is not moving. Vibhor, sorry for interruption. Your slide is not moving.
So might be some latency in network. Is it moving, sir?
No.
So let I check my connection once again.
Do one thing, you log out and you log in again. Then share your screen.
Sorry for some technical glitch, sir. Is it moving, sir? Hello?
No, it's not moving. Do one thing, just click on the slide which you want to explain.
Sir, this is moving from my side. Actually, I am explaining second slide right now. The outlines of my paper.
Okay. Okay, just give your presentation as it is. Go. Okay.
Outlines are introduction, literature review, proposed methods, data set description, disease classification, followed by experimental setups and conclusion. In introduction, the developing countries like large fraction of total population gain their livelihood from agriculture. Especially in India, around 54.6% of people from the total population depend on agriculture. Plant diseases are responsible for big losses in total crop productions worldwide. Most of the diseases in plants initially strikes the leaves of the plant. Later, its symptoms are evident on all parts of the plants. Traditionally, some methods like serological, DNA-based molecular methods are used in the laboratories for plant disease identification. Visually inspecting the diseases are error-prone and expert-oriented. The laboratory-based traditional tests are not in direct reach of the farmer. Therefore, it is a need of finding advanced ways to detect crop diseases through leaf images. Some advent of machine learning or deep learning techniques allows us with image processing to find the solution for such big problems. In this paper, method for detecting plant diseases through leaf images is presented, which is based on image processing and fine-tuned pre-trained models. For this purpose, we fine-tuned 9 pre-trained CNN models such as Efficient V7, DenseNet 201, VGG16, NASNet Mobile, Inception V3, Exception MobileNet, ResNet 50, and VGG19. These are fine-tuned to classify the diseases in crops, apple and urad bean. We have implemented the model, and the experiments are conducted on two data sets. One is benchmark plant village data set. It is publicly open. We have taken apple leaf images from this, and a self-prepared data set is also taken, also considered for our research. Urad bean crops leaf images were considered in this. The literature review considers the paper of Pradhan, Kumar and Mohan. They studied various pre-trained models and showed ability of transfer learning in identifying the illness of plants accurately and efficiently. In another study, modified DenseNet 169, VGG19 and NASNet mobile models by introducing some additional layers and evaluated them on potato leaf diseases. Authors found modified model promising and DenseNet model performed remarkable with 5-fold cross validation. In another study by et al. In 2022, they explored MobileNet and ResNet model to check diseases in apple leaf image datasets. The experiment revealed that MobileNet performed better and found less computational expensive. Here are some detailed survey and review paper done by us for plant disease detection using computational intelligence and image processing. The survey and literature review in detail has been done in these two papers. Now the proposed method, the workflow is presented in this picture. Is it visible, sir? Hello? Hello?
Hello?
Sir, is it visible?
Your slide is not moving. Continue. Hello?
Yes, sir.
Your slide is
Sir,
not moving.
I am once again resharing my slides. Try to resharing the slides once again. Sir, is it moving now? Hello?
Not moving.
Still not moving. Sir, what to do now, sir? Sir, I have shared successfully the slides.
Okay. So explain your work step by step.
Yeah, I was at the proposed method. This entire method initially starts with image acquisition. Here we had collected two data sets. One was plant village and another is self-created data set that is directly captured from the fields under the supervision of experts. uh, Image dataset have been prepared in this way. Image pre-processing, image augmentation is being applied to increase the size of data for better generalization, and the dataset splitting strategies further is used to find the training and test sets for training and validating or testing, evaluating the models. Dataset 1, which is DB1, it consists of 3,171 images of RGB channels. The size of these images were 256 into 256. It is a very popular and benchmark data set. The second data set, which was emphasized in this work, is DB2, the database of urad bean leaf images, which was prepared through capturing the images from smartphone directly from the fields at the location of Muradabad, Uttar Pradesh, in real-time conditions. So we have finally considered two data sets. One was under the control, which is laboratory conditions, the plant-based data set, and another is considered in real-time conditions. The Uradwin genotype KUG479 are captured, are used in this research work, which consists of three categories of diseases: CLS, Cercospora leaf spot, yellow mosaic disease, YMD, and Uradwin leaf crinkle diseases. The samples are given here, and small description with the number of images in the both the data sets are given here. Further, in this next table, crop diseases and their disease types with their symptoms are given. For disease classification, we fine-tuned 9 pre-trained models over Apple and Uradwin datasets. Transfer learning is the process of reusing pre-trained model on new similar type of problems. Therefore, machine exploits knowledge gained from a previous task to improve generalization about another. Fine-tuning is the process of taking a pre-trained machine learning model and further training it on a smaller targeted data set.
Okay, Vibhor, thank you so much for the presentation. Thank you so much.
Sir, any questions, sir?
No. You can end your presentation.
Before you can leave now.
Hello, good afternoon, sir. This side, Dr. Yogesh. Ayan, sir, all presentation are done or some pending?
Sir, actually, two of the participants have not responded to the calls. Earlier, they said that they will be joining on their time, but still they have not joined the session. So we are calling off the meeting.
Okay, no problem.
As the time has exceeded from 12:40, because this session was up to 12:40, so it has the time has been exceeded. So we are calling off the meeting.
Sure. Okay.
And thank you so much, sir, for coming, Dr. Yogesh, sir, and thank you, Saurabh, sir, for being a session chair or for sparing precious time with us. Thank you so much. Thank you, Yogesh,
Thank
sir.
you, Allah, sir.
Thank you.
You.